ATP synthase is an important enzyme that provides energy for the cell to use through the synthesis of adenosine triphosphate. ATP is the most commonly used energy currency of cells from most organisms. It is formed from adenosine diphosphate and inorganic phosphate, and needs energy. The overall reaction sequence is ADP plus Pi ATP, where ADP and Pi are joined together by ATP synthase. Energy is often released in the form of hydrogen ions, moving down an electrochemical gradient, such as from the lumen into the stroma of chloroplasts or from the intermembrane space into the matrix in mitochondria. Structure: located within the mitochondria, ATP synthase consists of two regions. The FO portion is within the membrane. The F1 portion of the ATP synthase is above the membrane, inside the matrix of the mitochondria. The nomenclature of the enzyme has a long history. The F1 fraction derives its name from the term fraction 1, and FO derives its name from being the oligomycin binding fraction. Oligomycin, an antibiotic, is able to inhibit the FO unit of ATP synthase. These functional regions consist of different protein subunits refer to tables. F1 ATP synthase structure. The F1 particle is large and can be seen in the transmission electron microscope by negative staining. These are particles of 9 nm diameter that pepper the inner mitochondrial membrane. They were originally called elementary particles and were thought to contain the entire respiratory apparatus of the mitochondrion, but, through a long series of experiments, Ephraim Racker and his colleagues were able to show that this particle is correlated with AT-base activity in uncoupled mitochondria and with the AT-base activity in submitochondrial particles created by exposing mitochondria to ultrasound. This AT-base activity was further associated with the creation of ATP by a long series of experiments in many laboratories. FO ATP synthase structure. The FO region of ATP synthase is a proton pore that is embedded in the mitochondrial membrane. It consists of three main subunits A, B, and C, and six additional subunits, D, E, F, G, F6, and 8. Binding change model. In the 1960s through the 1970s, Paul Boyer developed the binding change, or flip-flop, mechanism, which postulated that ATP synthesis is coupled with a conformational change in the ATP synthase generated by rotation of the gamma subunit. The research group of Johnny Walker, then at the MRC Laboratory of Molecular Biology in Cambridge, crystallized the F1 catalytic domain of ATP synthase. The structure, at the time the largest asymmetric protein structure known, indicated that Boyer's rotary catalysis model was, in essence, correct. For elucidating this, Boyer and Walker shared half of the 1997 Nobel Prize in Chemistry. Jens Christian Skar received the other half of the chemistry prize that year for the first discovery of an ion-transporting enzyme, now K ATPase. The crystal structure of the F1 showed alternating alpha and beta subunits, arranged like segments of an orange around an asymmetrical gamma subunit. According to the current model of ATP synthesis, the proton motive force across the inner mitochondrial membrane, generated by the electron transport chain, drives the passage of protons through the membrane via the FO region of ATP synthase. A portion of the FO rotates as the protons pass through the membrane. The C-ring is tightly attached to the asymmetric central stalk, which rotates within the alpha-3 beta-3 of F1, causing the three catalytic nucleotide binding sites to go through a series of conformational changes that leads to ATP synthesis. The major F1 subunits are prevented from rotating in sympathy with the central stalk rotor by a peripheral stalk that joins the alpha-3 beta-3 to the non-rotating portion of FO. The structure of the intact ATP synthase is currently known at low resolution from electron cryomicroscopy studies of the complex. The cryo-EM model of ATP synthase suggests that the peripheral stalk is a flexible structure that wraps around the complex as it joins F1 to FO. Under the right conditions, the enzyme reaction can also be carried out in reverse, with ATP hydrolysis driving proton pumping across the membrane. The binding change mechanism involves the active site of Ai² subunits cycling between three states. In the open state, ADP and phosphate enter the active site. In the diagram to the right, this is shown in red. The protein then closes up around the molecules and binds them loosely the loose state. The enzyme then undergoes another change in shape and forces these molecules together, with the active site in the resulting tight state binding the newly produced ATP molecule with very high affinity. Finally, the active site cycles back to the open state, releasing ATP and binding more ADP and phosphate, ready for the next cycle of ATP production. Physiological role: like other enzymes, the activity of F1-FO ATP synthase is reversible. Large enough quantities of ATP cause it to create a transmembrane proton gradient. This is used by fermenting bacteria that do not have an electron transport chain, and hydrolyze ATP to make a proton gradient, which they use for flagella and transport of nutrients into the cell. In respiring bacteria under physiological conditions, ATP synthase, in general, runs in the opposite direction, creating ATP while using the proton motive force created by the electron transport chain as a source of energy. The overall process of creating energy in this fashion is termed oxidative phosphorylation. The same process takes place in the mitochondria, where ATP synthase is located in the inner mitochondrial membrane. Evolution of ATP synthase: the evolution of ATP synthase is thought to be an example of a modular evolution during which two functionally independent subunits became associated and gained new functionality. This association appears to have occurred early in evolutionary history, because essentially the same structure and activity of ATP synthase enzymes are present in all kingdoms of life. The F-ATP synthase displays high functional and mechanistic similarity to the V-AT base. However, whereas the F-ATP synthase generates ATP by utilizing a proton gradient, the V-AT base generates a proton gradient at the expense of ATP, generating pH values of as low as 1. The F1 domain also shows significant similarity to hexameric DNA helicases, and the FO domain shows some similarity to H+- powered flagellar-motor complexes. The I plus or minus 3I squared 3 hexamer of the F1 domain shows significant structural similarity to hexameric DNA helicases. Both form a ring with threefold rotational symmetry with a central pore. Both have roles dependent on the relative rotation of a macromolecule within the pore. The DNA helicases use the helical shape of DNA to drive their motion along the DNA molecule and to detect supercoiling, whereas the I plus or minus 3I squared 3 hexamer uses the conformational changes through the rotation of the I cubed subunit to drive an enzymatic reaction. The H+ motor of the FO particle shows great functional similarity to the H+ motors seen in flagellar motors. Both feature a ring of many small alpha helical proteins that rotate relative to nearby stationary proteins, using H+ potential gradient as an energy source. This link is tenuous, however, as the overall structure of flagellar motors is far more complex than that of the FO particle and the ring with circa 30 rotating proteins is far larger than the 10, 11, or 14 helical proteins in the FO complex. The modular evolution theory for the origin of ATP synthase suggests the two subunits with independent function, a DNA helicase with AT base activity and H+ motor, were able to bind, and the rotation of the motor drove the AT base activity of the helicase in reverse. This complex then evolved greater efficiency and eventually developed into today's intricate ATP synthesis. Alternatively, the DNA helicase H+ motor complex may have had H+ pump activity with the AT base activity of the helicase driving the H+ motor in reverse. This may have evolved to carry out the reverse reaction and act as an ATP synthase. ATP synthase in different organisms: human ATP synthase. The following is a list of humans' genes that encode components of ATP synthesis: ATP5A1, ATP5AL1, ATP5B, ATP5BL1, ATP5C2, ATP5D, ATP5E, ATP5F1, ATP5G1, ATP5G2, ATP5G3, ATP5H, ATP5HP1, ATP5I, ATP5J, ATP5J2, ATP5L, ATP5L2, ATP50, ATP5S, ATP6, ATP6 April 1st, ATP6 April 2nd, ATP-SBL1, ATP-SBL2, MTATP6, MTATP8, plant ATP synthase. In plants, ATP synthase is also present in chloroplasts. The enzyme is integrated into thylakoid membrane. The CF1 part sticks into stroma, where dark reactions of photosynthesis and ATP synthesis take place. The overall structure and the catalytic mechanism of the chloroplast ATP synthase are almost the same as those of the mitochondrial enzyme. However, in chloroplasts, the proton motive force is generated not by respiratory electron transport chain but by primary photosynthetic proteins. Bovine ATP synthase. The ATP synthase isolated from bovine heart mitochondria is, in terms of biochemistry and structure, the best characterized ATP synthase. Beef heart is used as a source for the enzyme because of a high concentration of mitochondria in cardiac muscle. E. coli ATP synthase. E. coli ATP synthase is the simplest known form of ATP synthase, with 8 different subunit types. Yeast ATP synthase. Yeast ATP synthase is one of the best-studied eukaryotic ATP syntheses. And 5F1, 8 FO subunits, and 7 associated proteins have been identified. Most of these proteins have homologs in other eukaryotes. See also, subunits of ATP synthase, ATP synthase alpha-beta subunits, ATP synthase delta subunit, ATP synthase gamma subunit, ATP synthase subunit C, other, ATP10 protein required for the assembly of the FO sector of the mitochondrial ATPase complex. V. ATPase, oxidative phosphorylation, mitochondrion, chloroplast, electron transfer chain, proton pump, transmembrane ATPase, flavor protein, P. ATPase, rotating locomotion in living systems. References, McCarthy R.E. A plant biochemist's view of H+- ATPases and ATP synthesis. JEXP. Bile 172, 431 a €441. PMIDA 9,874,753 a, Fernandez Mora NH, Oda T, Blair PV, Green DE. A macromolecular repeating unit of mitochondrial structure and function. Correlated electron microscopic and biochemical studies of isolated mitochondria and submitochondrial particles of beef heart muscle. J. Cell Bile 22, 63 a €100 doi, 10.1083/JCB 22.1.63. PMCA 2,106,494. PMIDA 14,195,622 a. Gresser M.J., Myers J.A., Boyer P.D. Catalytic site cooperativity of beef heart mitochondrial F1 adenosine triphosphatase. Correlations of initial velocity, bound intermediate, and oxygen exchange measurements with an alternating 3-site model. J. Bile. Chem 257, 12,030 a €8. PMIDA 6,214,554 a, Nakamoto, R.K. Scanlon, J.A.B. Al-Shawi, M.K. The rotary mechanism of the ATP synthase. Arch. Biocham. Biophys 476, 43 a €80 doi, 10.1016/JAB. 2008.05.004. PMCA 2,581,510. PMIDA 18,515,057 a, ABCD During C, M. Trout B, Oster G. Rotary DNA motors. Biophys J69, 2256 a €67 doi, 10.1016 per second 0006-3495, 95, 80096-2. PMCA 1,236,464. PMIDA 8,599,633 a, AB Anthony Crofts. Lecture 10, ATP Synthase. Life Sciences of the University of Illinois at Urbana-Champaign, ABC Interpro Database, ATP Synthase, A.B. Martinez L.O., Jacket S., Steve J.P., Ectopic beta chain of ATP synthase is an apolipoprotein AI receptor in hepatic HDL endocytosis. Nature 421, 75 9 doi, 10.1038/Nature 01250. PMIDA 12511957 Villiers J., Palmer P., Subania V., AL Organization of the Yeast ATP Synthase F0, a study based on cysteine mutants, THIOL modification and cross-linking reagents. Biochim. Biophys. Acta 1458, 443 €56 doi, 10.1016/2 0005-2728, 000093-1. PMIDA 10,838,057 Devenish R.J., Prescott M., Ruku X., Negley P. Insights into ATP synthase assembly and function through the molecular genetic manipulation of subunits of the yeast mitochondrial enzyme complex. Biochim. Biophys. Acta 1458, 428 €42 doi, 10.1016/2 0005-2728, 000092X. PMIDA 10,838,056 External links, ATP Synthase a splendid molecular machine, well-illustrated ATP Synthase lecture by Anthony Crofts of the University of Illinois at Urbana-Champaign. Proton and sodium translocating F-type, V-type and A-type ATPases in OPM database, the Nobel Prize in Chemistry 1997 to Paul D. Boyer and Johnny Walker for the enzymatic mechanism of synthesis of ATP. And to Jen C. Scott, for discovery of an ion-transporting enzyme, now plus, K plus ATPase. Harvard Multimedia Productions site videos ATP synthesis animation, ATP synthase molecule of the month.